Assalamu alaikum dear students. Welcome to the course Principle in Animal Life 2. This is lecture number 37. The topic is Evolution in Action, Recent Research on Natural Selection. Discussing agenda for the topic will be Introduction, Case Study number 1, Case Study number 2, Lecture Outcome. After watching and listening this lecture, students will be able to explain the validity of the theory of natural selection in light of recent research. Introduction. This is the last topic of chapter 5. In that chapter, we discussed various concepts related with evolution. We primarily discussed the Darwin theory of natural selection, Darwin's theory of evolution by means of natural selection. Darwin in 1859 published a famous book entitled On the Origin of Species by means of natural selection. In that book, he proposed natural selection as a mechanism of evolution, and this theory brought a revolution in the field of biology, and ever since from time to time the researchers have undertaken studies to test this theory, to assess the validity of this theory, and now after 161 years of the publishing of that book and the presentation of that theory, we will assess or test the validity of this theory with examples that are on the offer in the surroundings nowadays. So the theory of evolution by natural selection is testable, means it can be tested. We can manipulate the environmental conditions for a certain population, and then we can assess whether this population adopts accordingly to these changing conditions or not. Indeed, researchers have documented heritable variations and differential reproductive success in a wide array of natural populations. Darwin mentioned in his theory that the organisms which have certain heritable features, heritable traits that makes them more adaptive to the prevailing environmental conditions, then such individuals will leave more offsprings, and such beneficial traits will be propagated in the population. The individuals carrying such beneficial traits will increase in number. This lecture summarizes two examples in which evolution by natural selection is observed in nature. Evolution is obviously a very slow process. It takes hundreds of thousands of years, but in our daily lives, we do have some examples or studies have been carried out. Those studies which were spread on a period of 30 or 40 years, and those studies found some examples which provide a clue to the validity of the Darwin's theory of evolution by natural selection. Means they validate that this theory is a sound theory and it has applications in the wild environment. Literally hundreds of other case studies are available involving a wide variety of traits and organisms. So to begin, let's explore the evolution of drug resistance, one of the great challenges facing today's biomedical researchers and physicians. Drug resistance is a phenomena where a particular drug which used to be effective against certain pathogens becomes ineffective. The pathogen somehow develops resistance against this drug, and even after the administration of that particular drug, the diseased condition is not cured. So it is obviously because of the fact that the pathogens which are responsible for the disease have developed resistance against that particular drug, and it's a dilemma, it's a headache for the biomedical researchers, because they are the ones who carry out research, who develop drugs after years of hard work and struggle, and all of a sudden when they come up with some hopeful results and they find some drugs, and after 3 or 4 years when they see the resistance from pathogens, then it feels like as if their 30 or 40 years of effort has been wasted. So now we will discuss the first example. How did Mycobacterium tuberculosis become resistant to antibiotics? Mycobacterium tuberculosis is the causative agent of tuberculosis or TB. The person affected with this bacterium suffers lung infection, and as a result fever is caused, there is coughing, sweating, weight loss, and death often results. In Europe and the US, TB was once as great a public health issue as cancer. Nowadays, as of 2020, the most debilitating conditions for biomedical researchers is cancer. How to get rid of this disease, how to find a cure for this condition. So once upon a time, the same headache was created for the biomedical researchers by this condition. They were unable to find a preventive measure, a preventive strategy, or develop a drug which is effective against this condition. Physicians were particularly alarmed because the strains of Mycobacterium tuberculosis responsible for the increase were largely or completely resistant to antibiotics that were once extremely effective. As already mentioned, that a biomedical researcher wastes years after year in finding a drug, a cure for a certain pathogen, and then once the pathogen develops resistance against that drug, then it just multiplies the problem. And the researcher will have to study everything right from the beginning to develop a new, more effective drug. How and why did the evolution of drug resistance occur? So what is the reason that a particular drug is effective against a certain pathogen, and then after some time the same drug becomes ineffective or less effective against the pathogen? So how the bacteria or the pathogens develop resistance against a particular drug? The case of a single patient, a young man who lived in Baltimore, it's a state in US, illustrates what is happening all over the world. So this case of the resistant bacteria was reported there, and you can assume that a similar kind of situation must have occurred elsewhere in the world or occurs elsewhere. So the man was suffering from fever and have a cough. Chest x-rays were taken, and the sputum analysis coughed up from the lungs revealed that the person is suffering from TB infection. He was prescribed doses of antibiotics for 6 weeks, followed by 2 weekly doses of antibiotic rifampin for an additional 33 weeks. So this was the treatment regime for the TB. And after 10 months, when the person returned, he was found TB negative, means no Mycobacterium tuberculosis was found in the sputum samples. So the doctor declared him as healthy and TB free. But after 2 months, the person returned again to the hospital, and he had same symptoms. They were a bit more severe. There was a fever, severe cough, and labored breathing, means he was facing difficulty in breathing. So once again it was the onset of TB for the second time. Despite being treated with a variety of antibiotics including rifampin, he died of respiratory failure 10 days later. So it was a dilemma for the doctors that once initially TB was reported, and then the prescriptions were made, the doses were taken accordingly, the patient recovered, but after 2 months of recovery, he again found himself in the hospital and was suffering from the same condition. So what had happened inside his body? Samples of fluid from his lungs showed that Mycobacterium tuberculosis was again growing actively there. Here the tests were negative after the initial prescriptions, and upon second visit after 12 months, the bacterial strains were again reported, and they resulted in his death. So it was a mysterious situation for the doctors, and they tried to find what has happened inside his body, which resulted in the recurrence of this disease, or whether he acquired these bacteria for the second time, or whether they were the reminiscence of the first infection. But this time the bacterial cells were completely resistant to rifampin. Initially rifampin was very effective against these bacterial strains, but on the second recurrence, this drug was found ineffective against those bacteria, because the bacteria had developed resistance against rifampin, and because of those resistant bacteria, they further proliferated. The symptoms in the patient died. So where did they come from? Where did those resistant bacteria came from? Either the patient acquired those bacteria for the second time, or some of the bacteria from the first infection evolved into mutant strains. So what had happened there? Could a strain that was resistant to antibiotic treatment have evolved within him? To answer this question, a research team analyzed DNA from the drug-resistant strain and compared it with stored DNA from Mycobacterium tuberculosis cells that had been isolated a year earlier from the same patient. So when the patient was first admitted to the hospital, these bacterium were isolated from his sputum and they were stored. And when the infection reoccurred after 1 year, bacterial samples were again collected and isolated. And when the patient passed away, those researchers, they analyzed and compared the two bacterial samples that were collected 1 year apart from each other. After examining extensive stretches from each genome, the biologists were able to find only one difference. So there was only one difference in the genome of the bacteria that were taken 1 year apart. And what was this change? This change was a point mutation in gene called Rpo. So in the resistant strains, this particular gene had mutated. It had a point mutation. The Rpo gene codes for a component of RNA polymerase. RNA polymerase is a complex, it is a group of enzymes, and this gene provides, its product provides one of the components of RNA polymerase. This enzyme, RNA polymerase, all of you know very well that it is responsible for transcription. It synthesizes messenger RNA from DNA, so it is essential to the survival and reproduction of bacterial cells. In this case, in the resistant strains, mutation occurred in this gene. What was this mutation? Cytosine of this gene at one position was replaced by thiamine, or it was modified to a thiamine, forming a new allele for the Rpo gene. So this missense mutation caused a change in the amino acid sequence of the RNA polymerase. The amino acid sequence of the RNA polymerase before mutation had serine at 153rd amino acid, but after mutation that serine became leucine, and because of this change in the sequence of amino acids, a change occurred in the shape, and the shape of RNA polymerase was slightly changed from its initial original form, and this rifampin drugs, its mode of action is that it binds with the RNA polymerase of bacteria and prevents the synthesis of RNAs or any kind of product from bacterial genome. But now there was no binding between the two, and therefore rifampin was no longer able to bind correctly with RNA polymerase and degrade it or prevent it from transcribing. So the RNA polymerase kept on transcribing genes from the bacterial genome, and the bacteria thrived and progressed with its pathogenic activities. Bacterial cells with the CT mutation continue to produce offsprings efficiently even in the presence of the drug, because the drug cannot bind efficiently to the mutant RNA polymerase. These results suggest the steps that led to this patient's death. So in this figure you can see the sequence of events which occurred and which ultimately resulted in the death of the patient because the mutation took place, cytosine was replaced with thiamine, and the patient was initially cured from the condition, but some of the bacterial cells had that mutation, and then those bacterial cells they thrived, they increased in number, and they were responsible for a reinfection for the second time, which eventually resulted in the death of the patient. Does the sequence of events illustrated in example 1? Now, initially our topic was to assess the validity of the natural selection, the theory of natural selection in light of current research. So now we will discuss the postulates of the Darwin's theory, and then we will try to find answers from this example for those postulates. One way of answering this question is to review Darwin's 4 postulates and test whether each one was verified. So his first postulate was, did variation exist in the population? So the answer is, due to mutation, both resistant and non-resistant strain of TB were present before administration of the drug. Most Mycobacterium tuberculosis population in fact exhibit variation for the trait. So there were variations for the Rpo gene. Some strains were not having the mutation, so the rifampin drug was effective against them, where other strains do had mutations in the Rpo gene, and rifampin drug was no longer effective against them. So variations were present, and there were one resistant bacterial cell per 10 raised to the power 7 or 10 raised to the power 8 cells. Means this variation was very, very small, negligible, but it was there. The second point is, was this variation heritable? Certainly yes, because initially there were very few bacterial cells which had this variation, this mutation in Rpo gene, but those bacterial cells carrying that mutation, they reproduced through binary fission, and they increased in their number, and they passed on this variation, this mutation to their new generations. So the researchers showed that the variation in the phenotype of the 2 strains from drug susceptibility to drug resistance, drug susceptibility in the initial case when the drug was effective, and drug resistance in the second case when the drug was not effective because of mutation in Rpo gene. So from drug susceptibility to drug resistance was due to variation in their genotype, because the mutant Rpo gene is passed on to daughter cells when a Mycobacterium replicates the allele and the phenotype it produces. Drug resistance are passed on to offsprings. Was there variation in survival and reproductive success? Yes. The bacteria which had mutation in the Rpo gene, they were more adaptable to the environment having rifampin, and they had greater ability to reproduce in those circumstances as compared to bacteria which had no mutation in the Rpo gene. So, were survival and reproduction non-random? When rifampin was present, certain cells, those with the drug-resistant allele, had higher reproductive success than cells with the normal allele. Mycobacterium tuberculosis individuals with the mutant RPOB allele had higher fitness in an environment where rifampin was present. The mutant allele produces a protein that is an adaptation when the cell's environment contains the antibiotic. So this study verified all four postulates and confirmed that evolution by natural selection had occurred. It is critical to note, however, that the individual cells themselves did not evolve. They could not mutate their genes in order to survive the antibiotics. So this mutation was not brought about by those bacteria in themselves. It was because of some event which proved beneficial for them. So natural selection does not acts on individuals, it acts on the populations. For example, if only one individual in a population changes, then we cannot say that this population has evolved. Majority of the members of that population must change to qualify the definition of evolution, and only then their gene and allele frequency will change. So this antibiotic resistance is a huge problem nowadays. In this figure you can see the patients which have ended in intensive care units in the hospitals because they had those bacteria and they were resistant to that antibiotic. You can see here the resistance was minimum in 1975 to 1990s, and negligible amount of patients were in ICU for resistant bacterial conditions, but then the cases spiked. The cases spiked in 1990s, and similarly the patients became affected with resistant strains, and their number in ICU wards increased, and if you can see, this graph is still on the rise. So these doctors are prescribing antibiotics for minor infections, for minor conditions, which is helping bacteria to evolve. So this was the first example. Here the change in the environment was man-made. The bacteria were initially thriving until antibiotic was prescribed. So this antibiotic was a change in their environment, and then they adapted themselves against this induced change. Now in the second example we will discuss a situation where the natural phenomena brought about a change. Why do beak sizes and shapes vary in Galapagos finches? So Galapagos finches were initially studied by Charles Darwin, and he hypothesized that an ancestral species migrated from the South American mainland and spread in different habitats on the Galapagos Islands, and they evolved into different species of finches, and they were primarily having differences in their beak structure, which was adapted to the type of food available on each island. So have biologists also observed evolution caused by natural environmental changes? The answer is yes. Very few cases are there where nature has induced a change in the phenotype, because such changes occurs at a slow and steady pace, and a lot of time is required to study them. Peter and Rosemary, they devoted 4 decades or 40 years of their lives. They studied the beak size, beak shape, and body size of the finches that were found on the Galapagos Islands. Remember, Galapagos is a group of islands. It is not a single island. So here we will discuss the finches that are found in one of its islands, and that island is Daphne Major. So the researchers, they analyzed, they captured, and they tagged the medium ground finches on that island. It is a small island equivalent to 80 football fields, and mark each one with a unique combination of colored leg bands for their identity. So the medium ground finch makes its living by eating seeds. Finches crack seeds with their beaks. Early studies of the finch population established that beak size and shape and body size vary among individuals. Means these are traits which shows continuous variation, and that beak morphology and beak size are heritable. Means the offsprings tends to have the same beak size and body size as that of their parents. More specifically, parents with particularly deep beaks tend to have offsprings with deep beaks. Now, what is the depth of a beak? So, suppose this is a beak. This is the beak of a bird. Now, this is the length of the beak. This is the length of the beak, and this is the depth of a beak. This. The breadth cannot be shown in 2D structure. So this is the depth of the beak, and this is the length of the beak. So large parents also tend to have large offsprings. Beak size and shape and body size are traits with heritable variations. They are passed on to their offsprings. So then all of a sudden the environmental conditions changed. In 1877, in the annual wet season, only 24 millimeters of rain was reported, which was well below the normal 130 mm range. So the rain, the area didn't receive a lot of rain, so it affected the vegetation, the plant life in that area. During the drought, few plants were able to produce seeds, and 84% of the medium ground finch population disappeared. Obviously, those finches, they depend upon the plant seeds, so when the plants were wiped off by the drought, it affected their immediate consumers, which were the medium ground finch population. So those finches also started to disappear. Now, two observations support the hypothesis that most or all of these individuals died of starvation. 38 dead birds were emaciated. Emaciated means abnormally thin or weak because of absence of food or illness. So because there was a shortage of food in that area, so 38 birds were reported dead, and those birds were showing symptoms of this condition. They were weak, they were starved because of the lack of food. None of the missing individuals were spotted on nearby island, and none reappeared once the drought had ended and food supplies returned to normal. So the researcher team realized that the die-off was a natural experiment. And in laboratory we usually manipulate certain conditions, but we have a control group, then we compare the control group with the variable group. Here in the natural settings, the natural was an experiment. The change in the environment was a variable, so the population of the birds and their morphology before the change and after the change was assessed. Before the change provided a control group, and after the change provided a experimental group. Instead of comparing groups created by direct manipulation under control conditions, natural environments allow researchers to compare treatment groups created by an unplanned change in condition. As already mentioned, that in laboratory we have a control group and a treatment group, but here in nature the change in environment they brought about an experimental group. So were the survivors different from non-survivors? Non-survivors means before the drought event, all the individuals before the drought event. So these two populations were compared, the ones that were present before the drought event of 1974, the population which was there and the population which was there. So these two populations were compared. Obviously there was a decrease in number, but was any change observed in any phenotypic characteristics? So on average the survivors tended to have much deeper beaks than did the birds that died. Why were deeper beaks adaptive? At the peak of the drought, most seed sources were absent, and the tough fruits of a plant called Tribulus cystoides served as the finch's primary food. Because of the drought, their primary food source was decreased in population. the drought drastically affected the growth of the plants whose seeds were consumed by the finches. So then when the population of those plants decreased, the finches searched out to find other plant seeds for their consumption, and they chose to feed on plants having seeds that were tough and not easy to break. So for such seeds, they needed beaks which had greater depth, because this greater depth provides strength to the beak, and they can break tough seeds with them. So the finches, after the drought, their beak depth increased, and it was because of adaptation to a new food source. There you can see the average beak depth increased after the drought. You can see here the increase as compared to this one. This shows the number of individuals before and after. So in 93, the environment of the Galapagos changed again over a 7-month period. Now this time there was much more rainfall as compared to 1974. So this greater rainfall again favored the conditions for the growth of the seeds which those birds initially fed on. So now what will happen? Now they doesn't need to have beaks with greater depths. So now, as an adaptation, the depth in the beak decreased. So you can see from those examples that the individuals adapted themselves to the prevailing environmental conditions. When the seeds had thin coats, they had beaks with less depth. When the seeds having thin coats become scarce, then they developed thick beaks. So because they were better able to harvest the small seeds, therefore the decrease in the depth of the beak took place. Now this is probably the most important sentence of this whole lecture. The take-home message from the data is that most traits are not inherently good or bad. So you can see from the second examples that initially the thin beaks were effective, but then because of the change which favored deep beaks, and then in 1980s, when the conditions changed, the environment once again favored the beaks with less depth. So the good or bad of a trait is determined not inherently but in light of the prevailing environmental conditions. A trait which is good for a specific environment may not be so good if the environment changes, and same is the case with a trait which is bad for a certain set of environment can be good and effective when the environment changes. This is the end of the lecture. Thank you.